Hello guys, welcome back to Excelsior Academy. So we were doing translation, and in that we have discussed about the initiation, like how the pre-initiation complex is being formed and then how elongation is carried out, correct? So this is just part of elongation only. So in the previous lecture, we have talked about the function or not, the role of EF2, if you remember, right? Elongation factor 2, we have talked about it. So what is the function of EF2? It brings your charged tRNA. It brings your charged amino acyl tRNA, right? And it masks the amino acid so that this amino acid is not getting involved in the chemical reactions present in the cytoplasm. Okay, so it is going to mask this amino acid and bring to the A site. So this is what we have discussed about the EF2, and it is also bound to GTP. Okay, if you remember in the previous lectures. Now, so further to the elongation, let's talk about the ribosome is also a ribozyme. Why? Because here we are going to discuss about the peptidyl transferase. So this we have discussed before also, that here the peptidyl transferase is not the protein. Okay, it's not a protein, hence it's not an enzyme. It's not a protein nature enzyme, but it is a ribozyme. And what are ribozymes? We have seen before also. They are like self-cleaving or self-catalyzing RNAs. So before we have talked about the introns 1 and 2, where they can act on themselves during the intron splicing. Similarly, in the ribosome, we have got the peptidyl transferase present at the P site, which is a ribozyme. Okay. Now, once the correctly charged tRNA has been placed in the A site and rotated into the peptidyl transferase center, the peptide bond formation takes place. So if you remember that A site, यहाँ पे tRNA आपका ऐसा रहता है, okay, it gets twisted to this rotation. If you remember, it is also known as accommodation around 70 degree, sorry, not 70 degree, by 70 angles from it gets rotated. Okay. So this reaction is catalyzed by RNA, which is your 23S rRNA component of the large subunit. Now, how do we understand that? Like, किसने हमें बताया कि राइबोजाइम ही है जो आपका कैटलिटिक काम कर रहा है? So all those crystallographic structures are there which have shown that without any proteins, okay, without any proteins, still the ribosome was able to carry out peptide bond formation. So what does this evidence say? It says that it is the RNA only which is catalyzing the peptide bond formation without any proteins. Okay. To support this, structural views of the large subunit of the ribosome from the prokaryotic species showed that there was no amino acid within 18 angstrom of the active site. तो जहां पे आपका P साइड का एक्टिव साइट है, जहां पे आपका पेप्टाइड बॉन्ड फॉर्मेशन हो रहा है, वहां पे there is no presence of the amino acid. So this was another structural evidence which proved that the any proteins are not required for your peptide bond formation. Only RNA was present. Okay, so I have got the figure also over here. Look it over here. Now in the figure, if you see carefully, you can see that this purple color things are proteins. Okay, and these gray ones are your RNA. Now, looking at this, this is your catalytic site. And what do you observe at the catalytic side? If you see in the surrounding areas, like over here this much, there is no proteins, right? Or no amino acid, you can say. If you zoom it and properly, if you check or measure the length also, you can see that around 18 angles from distance, there are no amino acid present. Okay. So from the figure also, it was clear that this is the actually 50S large subunit. Okay. So in that, they have seen that even though the proteins are involved, but at the catalytic site, there are no presence of proteins, so no presence of amino acids. So the catalytic site is entirely, the work is entirely done by the RNA. Okay. Is it clear? Are you able to follow?
Yes, ma'am.
Okay. Now the question is, कैसे आपका 23S RNA or the peptidyl transferase is able to catalyze the reaction? Okay. So the first thing is base pairing between the 23S RNA and the CCA ends of the tRNA. CCA ends of the tRNA in the A and P site positions, the amino acid, alpha amino acids of the amino cell tRNA to attack the carbonyl group of the growing polypeptide chain which is attached to the peptidyl tRNA. बहुत कंफ्यूजिंग लग रहा है, राइट? सिंपल भाषा में tRNA is having the amino acid, correct? And peptidyl transferase or the enzyme which is present over here. At the A side, this is present, and P side also you have got the another tRNA having the amino acid, correct? Which is having the chain of amino acid. Now, how this is catalyzing? So what we know is यहाँ का amino acid हटके यहाँ पे हो जाता है. So the bond is formed over here. Okay. So what does it says? It is the carbonyl group of the growing polypeptide attached to the peptide tRNA. So यानी कि यहाँ का carboxyl group और यहाँ का amino group is present, okay? And then they form a peptide bond. So आपका वहाँ पे CONH bond form होगा. Are you able to follow? So it means what? यहाँ पे जो amino acid है उसका amino group free है, okay? And C terminal attached है. यहाँ पे क्या है आपका? Carbonyl group attached है over here to this tRNA, P side tRNA. So the carbonyl group of this tRNA present at the P side is going to attack over here to the amino group over here of this A side tRNA, and hence it forms a peptide bond. So this kind of these interactions are also likely to stabilize the amino acid tRNA after accommodation, and such mechanisms are known as entropic catalysis. So these enzymes brings by bringing the substrate together in a manner that will help in the catalysis. अभी आपको और clear हो जाएगा once you see the figure. Now look it over here down. Okay. Now this much is clear, right? This is your amino acid tRNA, correct? Now can you see over here how it is attached? Yes, are you able to follow? Can you see here? ये आपका क्या था? यहाँ पे हम कैसे बोलते हैं? यहाँ पे आपका 3' में CCA है, correct? So this is your CC adenine group present. Okay. Now this is present at the 3'OH. If you remember, we have talked either it can be 2'OH or 3'OH in the previous lectures, right? Now here let us say that this is 3'OH. 3'OH carboxyl group से attach है. If you see, ठीक है, carboxyl group से attach है आपका amino acid. This is some side chain, and here your amino group is free. Is this much clear? This structure is clear of amino acid tRNA.
Yes, ma'am.
Okay, now coming over here. So this is a peptidyl tRNA. Again here, can you see CCA? CCA, adenine is there. Again, it is at the carboxyl group. Okay. Now what we have said is, it is the carbonyl group which is attacking. Okay. Now let's see how it is being doing. It has been proposed that 2'OH of the P side tRNA may act as proton shuttle. So 2' कौन सा हो गया आपका यहाँ पे? Can you see? 2'OH of your P side tRNA. Now this is one is acting as a proton shuttle. So in this model, 2'OH, okay, it is going to donate hydrogen to the 3'OH. Can you see? It is going to donate donate hydrogen to the 3'OH of the peptidyl tRNA. तो यहाँ पे क्या होगा? इसका H हटके यहाँ चला जाएगा and accepts a proton from attacking alpha amino group of the amino acid attached to the A side tRNA. Look it over here. So यहाँ पे वो क्या कर रहा है? When you have dotted line like this, it means those are hydrogen bonds. ठीक है? Those are hydrogen bonds. Now, what is happening? 2'OH का जो H था वो आपने दे दिया किसको? 3' को, correct? And amino से आपने H ले लिया. That's why if you see 2'OH जो है, this H is highlighted as red in color. It means it has taken from the amino group of the tRNA, amino acid tRNA. Are you able to follow this? Right? And 2'OH का जो H था वो आपने 3'OH को दे दिया है. Okay? Are you able to follow, guys? दोबारा से बताती हूँ. See, just follow the यहाँ पे जो दिया हुआ है. First, what you see is that आपका 2'OH is there. Now this H is making weak hydrogen bond with the 3 dash CO के साथ. Can you see this? This is hydrogen bond. And here the hydrogen bond is formed between the O and H. ये H कहाँ से आ रहा है आपका? Amino group of amino acid से आ रहा है. Is this much is clear to you guys?
Yes, ma'am.
Okay.
Yes, ma'am.
Now what is happening is you see the interactions, right? Weak hydrogen bond. Now this H जो आपका 2 dash का था, that has been taken up by the 3 dash. ये जो H है amino group का, वो आपने किसको दे दिया 2 dash को? So 2 dash is having H from the amino and this 3 dash is having H from the बाजू वाला 2 dash से. ठीक है? Now during this process, what had happened? The carbonyl has attacked the amino group. Oh, sorry, the amino group has attacked the carbonyl group. You can say like that. And these got free. तो COO bond था वहां पे, वो break होने के बाद the bond is found between here with the NH. Because यहाँ पे फिर electrodeficient हो जाता है. So in order to maintain that, the bond is formed. So covalent bond is formed, if you see. And hence this results in the peptide bond formation. Now look at the diagram again. Are you able to see? Yes. Carbonyl CO. Carbonyl has attacked over the 2 dash OH. ठीक है, वहां पे हाइड्रोजन बॉन्ड हुआ. जब यहाँ पे ये ऐसे जुड़ जाता है तो O नेगेटिव आ जाएगा. O नेगेटिव आ जाएगा, so this will try to make a bond with this to satisfy its valency, right? अब यहाँ पे क्या हो गया? NH है, N का है 3 valency, so एक H है, एक यहाँ bond है. Now one is free. So this will try to attack over here to form a bond. So here CONH bond फिर बनेगा आपका. Yes. Should I repeat again? Able to follow? Clear है?
Yes, ma'am.
Okay, okay.
Yes.
Good. Okay. So just understand, see arrow तो हम कहीं से भी शुरू कर सकते हैं, but always remember that it is the most electronegative carbon which will start the attack on the hydrogen or any proton. Okay. तो इसलिए सबसे पहले ध्यान रखो CO का शुरू होगा. ठीक है, यहाँ से वो यहाँ जा रहा है, यहाँ से यहाँ, यहाँ से यहाँ. Then only this is attacking over here to balance it. Okay, तो ये साइकिल ऐसे जाएगा आपका. Okay. So that's how we understood that it is the RNA which is catalyzing and not the protein. Okay. Now, peptide bond formation initiates translocation in the large subunit. Now, what is translocation over here? इसका मतलब क्या है? That which is present on the A side should go to the P side, और जो P side में है उसको E side में जाना है. ठीक है. So all these movements which is happening within the ribosome is known as translocation. So obviously this is going to result in the movement of mRNA by 3 nucleotides or 1 codon, correct? So this process is known as translocation. Now look at the figure. So we have got the figure over here which says that you have got your tRNA here. तो अभी हमें ये तो पता है tRNA जो ऐसे आता था, it becomes like this during the process of peptide bond formation, right? Now what is happening? You can see that once this amino acid or the polypeptide chain from P is transferred to A, can you see the bend in the tRNA? Are you able to see the bend in the tRNA, which is resulting in the exposure of your factor binding center as well as this tRNA is shifting towards E, thoda sa. If you see, there is a twist also. थोड़ा सा bending दिख रहा है. Yes. Now this conformation will actually recruit your another elongation factor called EFG, which is again bound to GTP. Okay. So once your factor binding center gets visible, there your GTP will come and sit. This much is clear, guys? Yes. Is this much is clear? Yes, Tanvi, clear है यहाँ तक?
Yes, ma'am.
Okay. Now, once this elongation factor GTP comes and sits over here, it is undergoing hydrolysis. Okay, since it is bound to GTP is undergoing hydrolysis. So what will happen? Hydrolysis hoga to this is going to get change in the structure, conformational change hoga. So from here, this shape, it is going to become straight. Okay, you can see it over here. So GTP hydrolysis has resulted in opening up of the elongation factor G structure. Now this opening has resulted in shifting of your tRNA. So you can see that जो tRNA आपका A में present था वो चला गया आपका P में, और जो P में था वो चला गया आपका E में. And structural studies have shown that the structure of this elongation factor G, okay, it's a protein. It has got structure similar to your tRNA, amino cell tRNA, okay. So that's why it was easy for them to sit at the factor binding center, and upon GTP hydrolysis, it was able to stretch out. And this stretching out resulted in pushing the A side tRNA to P side and P side tRNA to E side. Now, if you see, due to this opening of the structure, okay, pushing of the EFG resulted in proper alignment of your tRNA. Can you see it? अभी हमने कैसे देखा था कि अगर आप यहाँ पे ध्यान दो, यहाँ पे आपका tRNA ऐसे है, यहाँ पे amino acid है, correct? Now once this was pushed, it goes back to its structure here like this. So now this tRNA has become your P side tRNA or the peptidyl tRNA, and free tRNA has been exit out. Okay. So you can see this is also removed, this is also removed. So of course, any elongation factors, GDP means it has got less affinity. So it gets released. And here aapka ready ho gaya. Ab now what is the next step? So in the elongation now another charged tRNA will come. It will sit over here first like this, and then later it will twist in order to accommodate the tRNA. This will be transferred over here, and the same entire procedure will be repeated, where the function of EF2 and EFG is taking place. Is the entire figure is clear to you guys? This is really very important because elongation में इनका role बहुत important है, EF2 का और आपके G का, AFG का. So let's see the figure once again. So you have got the classic state when amino cell tRNA, okay, the peptide bond formation is taking place. It results in the hybrid state. Now hybrid state means where it is bending so that it can undergo peptide bond formation. The factor binding center is free that time. Okay, so that time elongation factor G bound to GTP gets recruited and sits at the factor binding side. एक बार जब यहाँ पे hydrolysis होगा of GTP, it is going to take the space of the A side, pushing the A side tRNA to the P side and the P side tRNA to the E side. So E means it is going to exit, it is going to leave the ribosome. Now it is free of any polypeptide chain, so it is free, it will leave the ribosome, and this tRNA will get into the P side position. After hydrolysis, the elongation factor G, it has got less affinity, so it is going to get removed from here, and your this initial ptRNA is also getting removed. Now this is ready for another round of amino acids. So another charged amino acid tRNA will come, sit with the help of the EF2 and with the help of the AFG, it will be shifted again here. Now if you see, that is what they have located in the mRNA case also, like just to highlight over here, they have shown that this first one amino acid tRNA, A side tRNA का codon yellow में दिखाया, फिर purple में दिखाया. So after this thing, after this EFG hydrolysis, you can see that A side में जो आपका amino acid codon था वो अभी आपका P में चला गया है, और जो purple जो P में था वो आपका E में चला गया है. So during this translocation, it is not only the tRNA which is getting shifted, it is also your mRNA which is getting shifted. ठीक है, so एक codon आपका shift हो जाता है. Okay. So this is here what they are saying is, as I have mentioned, how does EFG interact with the A side of the decoding center so effectively? So the answer is that they have got very similar structure. Okay, so your EFG, tRNA, they all have got similar structure to your factor binding site, which is present at the A side. And since they are made up of proteins and your tRNA is RNA, so they are actually giving a good example of molecular mimicry. यानी कि क्या उसका structure वो mimic कर रहे हैं, even though they are made up of proteins. Elongation factors 2 and G, they are made up of proteins, but they are going to mimic the structure. Okay, they are just going to copy the structural structure of your tRNA, and hence it is able to effectively carry out the elongation of the translocation. Is this much clear, guys? Akshay, able to follow?
Yes, ma'am.
Okay, okay. It's very simple. It's just that you just need to remember many stuff. Now, EF2 और GDP, उनको अभी दूसरे राउंड के लिए भी रेडी होना है, करेक्ट? Now, who is going to help them in getting bind to GTP again and removing the GDP? So वो काम कौन करेगा आपका? EFTS, elongation factor TS. So if you see over here, this yellow GDP is EF2. GDP हट के आपका EFTS आ गया है. Now EFTS is bound to your EF2. Once GTP is coming, your EFTS gets removed, and now your EF2 is ready with another GTP to mask the or recruit the amino acyl charged tRNA for the next round. Clear? Simple. आपके EF2G का जो GDP है उसको change करने के लिए from GDP to GTP, that work is done by the elongation factor TS. Simple. Okay. Now, a cycle of peptide bond formation consumes 2 molecules of GTP and 1 molecule of ATP. Now, who can tell me this answer? Yes, Tanvi, would you like to try? कहाँ पे हमने ATP use किया and कहाँ पे हमने GTP use किया to make one peptide bond formation? Would you like to try?
Ma'am, one molecule of GTP was used during the process when it get attached to that factor binding center, and then
हाँ, but वो factor का नाम क्या है? Okay, Akshay, would you like to try?
One GTP is used for that EFTU, and one GTP is used for EFG, and I guess the ATP is for the adenylation, the first process for charging the tRNA.
Correct, correct, correct. So this is correct. So you are using one molecule of ATP to charge your tRNA. So that is the first step where you have used amino acid is there, right? And ATP आता है, and that step is known as adenylation. And later this is getting transferred to tRNA, removing the AMP group, correct? So here one ATP molecule is being used, and as he said, 2 molecules of GTP, of course, EF2 bound to GTP is coming, where it is recruiting your this charged tRNA, correct? And then next EFG is there, which is helping in the translocation. So in short, what you can say is that one molecule of ATP is used for adenylation. Then one molecule of GTP, handwriting anyways, I hope you can understand, is used for tRNA recruitment. Correct? Then one molecule of GTP is for your tRNA translocation. Okay, correct, good attempt. Now, and the same thing, although the eukaryotic factors अलग से हमने discuss नहीं किया है, so EF2 जो prokaryotes में, it is known as small e EF1, and जो G था, it is small e EF2. So their functions are also remarkably same. तो यहाँ पे हमारा elongation factors or the elongation of the eukaryotic translation gets complete. Okay, both prokaryotes and eukaryotes, it is going to be the same. Okay, so I'll be stopping here now, and what is left is the termination. So there we are going to talk about the release factors. RF1, RF2, RF3 are there. So those different release factors are helping in the termination of the translation. So we'll discuss about them in the next lecture, that is tomorrow. Okay, yeah. Any other doubts are there in this session? It was very short and but precisely taken. Still, any doubts are there, Akshaya, Tanvi?
No, ma'am.
Okay, cool then.
No, ma'am.